Chapter 7, Genetic Combination and Transfer. The learning objectives for this topic are: define genetic recombination, contrast vertical gene transfer with horizontal gene transfer, explain the roles of an F factor, F plus cells, and HFR cells in bacterial conjugation. Describe the structures and actions of simple and complex transposons. Compare and contrast crossing over, transformation, transduction, and conjugation. The topics for this topic are genetic recombination, horizontal gene transfers among prokaryotes, transposons, and transpositions. So let's talk about what genetic recombination is. It's the exchange of nucleotide sequences between two DNA molecules. Often involves homologous segments of nucleotide sequences, and the common type is DNA molecule is nicked, transferred, and ligase reconnects the strands in a new combination. And recombinants are DNA molecules and organisms that have undergone recombination. So what does this all mean? Well, let's take a look at a diagram. So here are two homologous sequences. So we'll say a bacterium has picked up a new section of DNA. We'll go into how they do that in the following slides. So an enzyme comes along and snips part of the DNA homologous sequence of A, and then snips it on B, and they do a crossing over. In fact, it looks very, very similar to the crossing over in synapsis that happens during meiosis prophase 1. Here we go. We've got a new crossing over, new combination, in which we've had this part of A has switched over to B, and this part of B has switched over to A. And so we have recombinant A and recombinant B. In this situation, it would be recombinant DNA. If this were happening inside of an organism, instead of, say, in a test tube, we can do this in a test tube, the organism would be a recombinant. So how do bacteria get new genetic material so that they can become recombinants? Through horizontal gene transfer. Let's talk about the difference between vertical gene transfer and horizontal gene transfer. Vertical gene transfer happens from parent to child. You are a product of vertical gene transfer. You got half of your genetics from your father and half of your genetics from your mother. Comes together to make you. When you have children, then your DNA goes down to your children. In bacteria, vertical gene transfer would be a cell divides into two daughter cells. So once again, we're going from parent cell to daughter cell. Well, horizontal gene transfer is when you have genetics that transfer to organisms within the same generation. Animals don't do that. That would be like if I came up and shook hands with you and we transferred DNA. That just doesn't happen. But it does in prokaryotes specifically. We know it happens in bacteria. So let's take a look at the first one, which is transformation. Now, this was originally discovered by Frederick Griffith, and he was working on a vaccine for streptococcal pneumonia. There are two strains of Streptococcus pneumoniae, and one of them is called smooth, or S, because the colonies have a smooth appearance because the organisms have a capsule. So this makes the Streptococcus harder for the immune system to fight, because the capsule makes it so that white blood cells have a hard time phagocytizing them. If you take S strain and inject it into a mouse, the mouse is going to get pneumonia and die. Well, if you heat treat these, in other words, you kill the Streptococcus, but you still have the capsule, you inject it into the mouse, and of course the mouse lives because there's no live cells. Well, there's another strain of Streptococcus pneumoniae, and it is called R for rough because the colonies are rough and they don't have a capsule. Well, you inject that into a mouse, the mouse's immune system takes care of it before they can get pneumonia, and they live. Well, the next part of Griffith's experiment was to take living R strain, so this is Streptococcus that doesn't have a capsule, and heat-treated dead S strain, so dead cells that had a capsule previously, and he injected both into the mouse. Well, the mouse got pneumonia and died, and when they cultured the blood of the mouse, there were living S strain bacteria. Well, what happened? Did he discover zombie bacteria that have come back from the dead? Well, actually, no. What he did to find out what in the world was going on was to take the heat-treated cells, and he chopped up the DNA, and he mixed in living R strain, and he found that they transformed in the test tube. So what they figured out is, no, they weren't zombie bacteria. What was happening is the R strain bacteria were picking up pieces of DNA from the dead cells. They have transport proteins that can transport long segments of nucleic acids whole into the cell. Then recombination happens. Some of the R strains picked up DNA that coded for the capsule, and they started being able to produce capsules and became pathogenic. Kind of cool, huh? Now, bacterial cells that naturally do this, that can naturally produce that transporter protein, are called competent. And Streptococcus is one of them. We can cause transformation to happen in the lab by basically taking a cattle prod to the cells and making holes in them. We'll talk about that in the next chapter. So we can cause transformation to happen in non-competent cells by making them competent. So that's transformation, where a living cell can pick up naked DNA from its environment, usually from a dead cell. Next up, transduction. Let's talk about bacterial phages. Bacterial phages are viruses of bacteria. And here's how it usually goes. The phage lands on top of the cell, the bacterial cell, and injects its DNA in. It also injects an enzyme that will go through and chop up the bacteria's chromosome. Well, the bacteria can't transcribe that DNA anymore, so now it can only transcribe and translate the viral protein. The cell makes a whole bunch of parts for the phage and also makes viral DNA, and that viral DNA gets packaged into the head of the phage. Well, sometimes the phage makes a mistake and accidentally picks up bacterial DNA instead of phage DNA. If these were to go on, the ones that have viral DNA, they go through and do this kind of thing. They go through, chop up the DNA of the bacteria, make new phages. But this phage, which we call a transducing phage, does not have the genes to make more phages. It doesn't have the genes to chop up bacterial DNA. But this is kind of like a little spaceship on autopilot. So when it's released from the bacterial cell, it goes and grabs another bacterial cell and injects bacterial DNA in. This recipient cell lives, and it recombines with the transduced DNA, and we get a transduced cell with new characteristics. Kind of cool, huh? So transduction is when new DNA comes through the mechanism of a phage. Our last method of horizontal gene transfer that we're going to talk about is conjugation. Occasionally you will get a bacterial cell that produces pili, we call them conjugation pillus, that transfer DNA from the cell to the cell that it's grabbed a hold of. Now, this looks an awful lot like bacterial sex. The difference is the whole genome is not being transferred. So it's not really sex, but we do call it conjugation. Think of conjugal visits in prisons or whatever. This cell, which we call an F+ cell for fertility plus, and this cell is a fertility negative or an F negative cell. Here's what's really going on with these two cells. The F+ cell has a plasmid. Now remember that a plasmid is extracellular DNA. And plasmids always have what's called an F factor. It's the gene to code for conjugation pilus. So the plasmid is coding for this pilus. Once the F plus cell connects to an F negative cell, the plasmid replicates itself and it is passed through the conjugation pilus into the F negative cell, which is now an F plus cell, and now the F plus cell can make pili and pass the plasmid on. So it's more like a venereal disease. But instead of causing death and destruction, it can cause them to be superbacterium. Some plasmids only have the F factor, but an awful lot of the plasmids have resistance factors along with the F factor. Resistant factors are things like enzymes that make them resistant to antibiotics. So instead of getting sick with a venereal disease, now I'm superbacterium, I'm resistant to penicillin, thank you. You also have on plasmids bactericidal genes that can enable them to kill their neighbors in the area so that they can have the food. Generally, these genes that are carried on the plasmid benefit the prokaryote. So it's actually a good thing to turn into an F plus cell. Now, sometimes this plasmid will incorporate into the bacterial chromosome, and we call these HFRs, or high frequency recombinants. So what happens, we'll say this one has the plasmid incorporated here. When it grabs the F negative cell, the origin of transfer is now in the chromosome. So instead of trying to pass just the plasmid, it tries to pass the entire bacterial chromosome to the other side. Well, with fluid movement and whatever else is going on within the medium, these two cells will separate before the entire bacterial chromosome will pass over. So the recipient cell doesn't get the entire plasmid. So it doesn't turn into an F plus cell, nor does it turn into an RFR cell. But it does pick up some of the genes from the HFR cell. And so it will achieve horizontal gene transfer through conjugation. Now, I'll leave you to read in the textbook about how originally we figured out the order of genes on some bacteria through the use of high-frequency recombination cells. It's really quite an interesting read. On to transposons and transposition. Now, transposons have been called jumping genes because they move from spot to spot within a DNA sequence independently. And unlike recombination, they don't have to move to a homologous spot. A researcher, Barbara McClintock, discovered these jumping genes when she was looking at color of corn kernels, and she found that genes for the color turned on and off when these transposons inserted themselves or removed themselves. Let's take a look at the simplest type of transposon. That's where we have what's called an inverted repeat on either end and the gene for transposing, the transposase gene. So the inserted inverted repeats are palindromes of each other. In other words, if you take a look at the sequence here, you'll see that they are reverses of each other. So what happens is the transposon makes kind of a loop of itself. The transposase causes the transposon to make a copy of itself, and we end up with another transposon. So this is the simplest kind. We do have complex transposons that have not only the transposase gene, but also other genes. Oftentimes they carry resistance genes or R factors. In this example, it's a gene that makes the organism resistant to candamycin. So we're kind of keep an eye on transposons. So here's an example of a transposon jumping from one part of the genome to another. It causes a frame shift mutation. Sometimes it turns genes on, sometimes it turns them off. We've found transposons in all living organisms and including in viruses. So they're all over the place. And sometimes they can transpose themselves to a plasmid and when the plasmid crosses over through the action of conjugation, it moves to another cell. Interesting, huh? Well, that's it for this topic. Here's a table that goes over the different methods of horizontal gene transfer. And here's a reminder of what you needed to learn from this topic.